Swayam Prabha. Digital India. Educated India.
Okay, so as I told you, this week will be quite intense, and so I'm fulfilling the promise. And today, after the previous days where we had quite intense talks on the chassis, on orthogonality, or genetic tools, now today we are going to really get deep into one of the most important set of technologies in synthetic biology that have to do with genome editing, and genome engineering. So if the idea at the end is being able to program bacteria, to program cells, to do interesting things, obviously we need to have the tools to do that. And as the DNA at the end of the day is something that is written in a given code, we should be able to do it, and for doing that you need methodologies, and some of them are not trivial, and some of them I'm going to explain in some detail. And obviously this is a field that is developing very quickly, and the technologies that I will tell you today may not remain forever, but at least conceptually you have an idea of the type of, say, creativity that different people had to address this particular issue. Okay, so what is editing? So the term editing comes from, say, writing, and well, I'm sure that you all have been through having a text and having to enter some changes for improving the text or for changing the significance or for other purposes. So at the end, editing means having a text. In our case, of course, the text will be DNA, and then you may want to enter insertions, you may enter deletions, you may want to correct some typos, and you may want to execute some replacements. So all of them are signified here, okay? So in the case of engineering, or in the case of editing bacterial genomes, then the equivalent operations that you make as if you were editing a text would be this. You can delete out of an existing genome either genes or operands, entire operands. You can bring about an allelic replacement, so that means that you have a given gene somewhere and you have to make a variant of that gene for a given purpose. And you may also think on inserting heterologous DNA in the genome of your bacterium. Okay, so we have to go through some definitions that are important when discussing genome engineering and genome editing, okay? So some of them I'm sure that you know, others will have to revisit them. So genotype is the genetic constitution of an organism. Phenotype are the observable properties of an organism. A mutation is the alteration of the nucleotide sequence of a genome that can be spontaneously happen because of errors in DNA replication or because of DNA damage. Just remember that mutations happen all the time, and for instance in E. coli, even if we don't touch E. coli, we let it go as it goes, there is a spontaneous rate of mutations between 9 to the minus 9 and 9 to the minus 10. These are big numbers, and therefore, for the most part, we take for granted that in an invariable environment, the genome will remain stable. But in any case, there are always like some background mutations that are infrequent, but they do happen. So for isolation of mutants that go beyond this basal level, we need to do something about it. And in many cases, even before the times of genetic engineering and recombinant DNA, then microbiologists use mutagens. Then after mutagenesis, there was some selection, and if not selection, at least a screening to show or to reveal the presence of mutant cells. So mutagens are compounds or circumstances that may speed up mutations, for instance, X-ray or chemicals, and in the hands of microbiologists, also transposons. Selections are the conditions that allows growth on only mutant cells, and screening, unlike selection, is a method, is a kind of manual method to differentiate between wild type and mutant cells. Okay, so remember these things. Okay, so this will be a typical, flow chart of how to go in a mutagenesis experiment. So if you go through chemical methods, then you just use mutagens, then you select another screen and identify different phenotypes. And therefore, the idea in this case is that you determine what gene is doing what a posteriori. But in other cases, you may go into what is called reverse genetics. And in this reverse genetics, what you do is that you make a specific deletion in a gene and then look for a phenotype, okay? And therefore, one of the main tools that biology has to know what a gene is doing is to have a mutation in that gene and see what happens with the phenotype, okay? So it's a basic, biological concept. So in forward genetics, you mutagenize everything, and then you see what are the phenotypes that appear after that mutagenesis. In reverse genetics, you focus in one particular segment of DNA, in one particular gene, and then examine what are the resulting phenotypes. We are going to address today are different methods for engineering bacterial genomes, and you can distinguish different methodologies. There are methodologies that you may call classic, even though classic doesn't mean that you go 1,000 years. It goes, 10 years or so. And then you have some other methods that are advanced methods that are based on more contemporary, tools, such as recombination and CRISPR-Cas-based methods. And in these classes, you will have an overview of the different methods and how far you can get with each of them. Okay, so the first method that is classic, but classic means that it was published at the end of the 90s and early 2000s, is something that is based on homologous recombination. And well, this is a kind of advanced, I'm sorry that the lettering is a bit messy here, but the idea in this case is that you exploit homologous recombination. There is a phenomenon that happens in basically all biological systems. And how that works? Well, the system involves two plasmids, but there's something wrong with the framing of it. Okay, let's see if we can go there. Okay, so you have two plasmids here. One plasmid is a suicide plasmid. A suicide plasmid, as you may recall from previous talks, is one that can make it into the cytoplasm of a target bacterium, but it cannot replicate there. And then you have a second plasmid that expresses a nuclease that is called SCE1. And the reason and the structure of this plasmid you will see in a moment. Okay, so these are the two plasmids that we were talking about. You can forget the names of the plasmid, it's not important. Well, one of the plasmids is the provider plasmid. So the provider plasmid goes as follows. This is a cloning plasmid where you have either a multiple cloning site, as we talked the other day, or you have a modified cloning site that facilitates selection of insertion, because you have a laxed alpha trick that, as you may recall, reveals the difference between those variants or those clones that have inserted some DNA versus the clones that have not inserted DNA. And because it has an R6K or inner replication, then this plasmid only replicates in hosts that provide in trans the pi protein that is required for replication. Okay? So you have a cloning vector here that all it takes is being able to act as a receptor or recombinant here and also it has an original replication that is conditional for just one single host. And also it has an ORIT, so that means that it can be transferred into another bacteria through conjugation. Now, the other actor of this type of approach is this other plasmid, and there are very variants of one plasmid and the other, in which what you have is a conditional expression of a nuclease called 1SC1. And why we have this, you will see in a moment, but just remember that you have an enzyme here that cleaves something that is in the plasmid here, okay? So you express from this plasmid in vivo an enzyme that will cleave specific sequences that are in this plasmid. Now, what is the kind of business part of this plasmid? Well, it's very simple. You have Sil S, that is a transcriptional factor that responds to benzoate, controlling expression of this part, this biological part, that is the nuclease SCE1, okay? Now, this is what happens. So if the system, if the cells are, are not exposed to the inducer, then expression of SC1 is off. But when there's benzoate or 3-methylbenzoate, then you get expression of this enzyme there. Okay, now, let's suppose that you want to delete a segment in the genome here. Okay, you have some genes here, and then, for instance, you have 7 genes. It can be 1, 2, 3, or 40. It doesn't matter. And then you want to delete this region from the chromosome of your favorite bacterium. How do you do it? Well, the first thing that you have to do is to identify some flanking sequences at the sites of the segment that you want to delete. And for an optimal homologous recombination, the size of these fragments should be in the range between 500 and 800 base pairs. So you take a look to the sequence and you identify a DNA that, or the sequence of DNA that happens to be exactly upstream of what you want to delete and downstream that you want to delete. And then you have like 2 targeting sequences of homology, TS1 here and TS2, okay? And this is completely general. You have to do that in every genome. Now, second part. These regions of homology have to be amplified with PCR or by other methods, and they have to be created as segments of DNA that can be cloned somewhere else, okay? So these earlier regions that you had flanking, this long region here, the region 1 and the region 2, have to be produced as PCR fragments, okay? So now what you do is a PCR reaction that I will not get into the details, so that you join in vitro this segment of DNA here with this segment of DNA there. And at the end what you create is this other segment of DNA where you have a kind of artificial, say, conjunction of this segment that was originally here and the segment that was originally there, okay? So now you kind of figure out the situation here, and then, well, you make your PCRs and make sure that everything goes as it should. You have to, for making sure that you can later recognize where the deletion is, you have to think also on primers that will amplify the region of interest before and after the deletion, okay? Okay, so, and then you generate references of sizes of what you should get before and after the deletion here, okay? So you have it there. Now, here comes the interesting part. You now clone this fuse segment of DNA where you have a bit of the region upstream and a bit of the region downstream. You clone it in the plasmid that has SCE1 sites and cannot replicate unless it goes into a specialized strain that has the pipe protein for this original replication. But at the same time, it can be mobilized because of this ORIT. Okay? Well, in some cases, the cloning is simplified because there's this alpha complementation, and therefore identification of the right clones is very easy. But in any case, at the end, what you have is this construct. So this construct contains the left part of the deletion in the genome and the right part of the deletion in the genome there. Now, what you do is that you take this plasmid and you enter it in your destination strain. I put it here P. putida, but it can be any other bacterium. It can be E. coli, it can be any other gram-negative bacterium, for instance. And then what happens is, whether you introduce this plasmid through electroporation or through mating or through conjugation, at the end, if you select for kanamycin, then you are selecting for formation of cointegrates. So why is that? Because the only way this plasmid can survive inside the target bacterium is by integration of this plasmid through homologous recombination, either through this site or through this other site there. This is what happens. If you select for kanamycin, then this plasmid necessarily had to be integrated into the chromosome, because it cannot live as an autonomous plasmid, but then the whole, say, plasmid structure gets recombined either here or here, and therefore you form a so-called a cointegrate. And this is dependent on homologous recombination that itself depends on a mechanism that is based on this protein that makes the recombination. Obviously, you have a region of homology upstream and another region of homology downstream. The plasmid can be integrated either upstream or downstream. Is something that you can do. Okay, so one way or the other, once the plasmid gets integrated into the chromosome, then you get structures of this sort. Okay, so if you, when you have a recombination here, then you start integrating there, but then it continues like that, and then it continues the whole thing, and then it continues like that. It's like you have a circle that gets integrated into a site of the chromosome, and then you have a so-called cointegrate, right? So this would be the structures of what you have there. And how do you know the structures that you have in the chromosome? Because as you have already some primers that target here or here, then you can really distinguish in a gel, by making PCR, what type of cointegrates you have obtained in the clones that are resistant to kanamycin after executing the procedure. Okay, just as a detail, important detail, just remember that this is integrated into the chromosome, okay? And it happens that you have here SC1 sites, okay? Now, here comes the next important thing. The second plasmid comes into play, and this second plasmid expresses conditionally, as I said, this nuclease that is called SCE1. And you can introduce this plasmid, again, through electroporation or through mating into the recipient strain, in such a way that now you have the chromosome of your bacterium here with a cointegrate at one side or at the other side, and then if you induce this plasmid with one SCE1, then the enzyme will cleave the cointegrate either here or here, but at the same time that it cleaves the sites, it will kill the chromosome as well, okay? So because as you have just these single sites in the whole chromosome, if you cut the chromosome at these locations, then the chromosome is cut. And if the chromosome is cut, then cells will die, okay? So this is the kind of blow-up of what I told you before. So you have a cointegrate here. Keep in mind that this cointegrate is in the whole chromosome, so this will continue like this, the rest of the chromosome will continue like this, and if you cleave the chromosome, then the chromosome will be destroyed and will not replicate any longer. So because you create with this enzyme single strand breaks. So why we use this SC1 enzyme? Well, we use it because this enzyme is very rare. It hardly ever, you can very unusually find these type of sites in the chromosome of any bacterium. Okay, so the only way to avoid killing is that this sequence here recombines with this sequence here. Okay, so you have two sequences that are identical here and here, and then the way to maintain the structure of the chromosome without being subject to deletion or to the action of SC1 is if this TS1 recombines with this STS1, or if this TS2 recombines with these two ST2. Now, if this TS1 recombines with this TS1, then all you do is that you eliminate the plasmid insertion. But if TS2 recombines with TS2, then what happens is that then the entire segment between this TS2 and this TS2 will be deleted, and then what you have at the end is a situation where the intervening region is altogether deleted here, okay? So chances is that once you have the cointegrate and you cleave the cells, cleave the DNA with this enzyme, then either you recover what you had at the beginning, or you just eliminate the segment that you wanted to eliminate at the beginning, okay? So that's actually what happens, and therefore chances are that you will have 50% recovery of the wild type and 50% recovery of the deleted string. That is what you wanted to have at the end. Okay, so how can you distinguish one from the other? Well, again, through PCR. Once you have these clones treated with the enzyme, you induce this SC1, then you can distinguish very clearly the wild type situation versus the deleted situation. The wild type situation with some primers will give you a big band. The deleted situation will give you a much smaller band when you amplify the chromosomal segment with the same primers. Okay, so in this case, you can see it there very, very clearly. And then, well, you can now start making some confirmations. For instance, trying to amplify the region that you wanted to delete with internal primers, and you don't have any signal. That's a good indication that you will not have any, that you don't have any segment in between. If you do the same with the wild type, obviously you will have all these segments here that will be amplified. But if you do it with primers that go upstream and downstream of the region of interest, then you find nothing in between because there's nothing to amplify in between. Okay, so then there's another step, is that once you get your deletion, you have to eliminate the plasmid that encodes PSW1, encodes SC1, that is the enzyme that cleaves the genome, okay? And then that's a kind of fastidious part, and all you have to do is to grow the cells over and over and over and over until you lose the resistance that is present in this plasmid, okay? So you enter a plasmid to execute this cleavage of the chromosome, but then you just eliminate the plasmid through dilution of the or subculturing of the cells under conditions of no selection, and then cells eventually will lose the plasmid spontaneously, okay? So, well, this may look like a complicated procedure, and to some extent it is. You're required to handle some macrobiological techniques, and, well, the protocol involves the cloning of the sequences in the suicide cloning vector. That takes some time. Cointegration is very simple. Transformation is very simple. Induction of CY enzyme is very simple. Deletion confirmation is very simple. And then curation, sometimes it takes time. Well, although theoretically the procedure is effective 50%, because you can have the recombination going in one direction or in the other, in cases where the deletion incorporates or produces some type of toxic phenotype, then the ratio between the two might differ, and normally it differs against our interests, and this is something that you have to live with, but you know, it's not too bad. So if the mutation does not give you a bad phenotype or a phenotype that weakens the growth of the cells, in general you have indeed a 50% efficiency of the method. Okay, so in terms of the timing, and you know, you will see that timing is important, then getting one mutation may take around 2 weeks, okay? That if everything works. If the mutation is useful, and I mean the mutation is easy, then you can have it in about 10 days, but you know, there is no way you can do it in just a few days. You have to really spend some time, between 2 and 3 weeks, to get the mutation, the deletion mutation that you want. And sometimes you have the time, and sometimes you don't have the time. And well, this is good enough, but we can do better. Okay, so well, I have to say that this system of generating mutations through recombination is still the most common way of getting mutations in the genes, in the genomes of many bacteria, because even though it may be fastidious and time-consuming, it uses to work very well under many circumstances. Okay, but in any case, sometimes you want to make more mutations, and you want to do it faster and everything, and well, one really can do better. Okay, so and therefore new systems have been developed, and well, apart of being time-consuming, another big problem or another big limitation is that these systems are not multiplexable. So that means that you have to get one mutant at a time. Okay, I make one mutant and another mutant and another mutant and another mutant and another mutant. If you remember yesterday or a few days before yesterday, we made, for instance, a genome of Pseudomonas putida that had been deleted for about 30 genes. Okay, so these mutations were entered with the method I just told you, and making this strain took nearly 2 years, because you have to go making one mutation, 2 weeks. Then you have to prepare for the next mutation, another 2 weeks, and another 2 weeks, and another 2 weeks, another 2 weeks. So even though at the end what you have is very good materials, but the fact is that it's very time consuming. And so ideally, we would like to have a system that you can, mostly in cases where you want to make many mutations at the same time, you want to have a system that is multiplexable, that you can do many of these at the same time. Okay, so one way to go, and this is something that at some point was a big breakthrough, was another technology that is called recombinering. Okay, so it's not the same recombination that recombinering. Recombination deals with big chunks of DNA that you need for recombination. In the case of recombinering, as you will see in a moment, you require just small segments of DNA that are homologous in the target and in the in the target region of the genome for making this happen. Okay, so recombinering, instead of using homologous recombination, it uses phage-derived homologous recombination and proteins that facilitate homologous recombination in a donor, in a strain, in a chassis, in a recipient, in such a way that these homologous protein recombinations coming from phages act on shorter, much shorter DNA sequences. Okay, so this paper that is mentioned here is year 2000, early 2000s. Well, what it is the following. You, instead of having to clone a gene and to having all these systems that you need to conjugate and then you have to eliminate and all types of things, what these people develop is a procedure through which you can get mutations by simply using PCR fragments. Okay, so these PCR fragments can be produced in vitro, and they may carry antibiotic resistances. And to have integration of these PCR segments in your favorite say site, you need very small homology sequences at the end. So you need to have these huge sequences, but something very simple. Then if this PCR fragment is constructed flanking an antibiotic resistance, then what you do is that you take this PCR fragment, you transform that into your favorite bacterium, and then you just select for the phenotype of antibiotic resistance. And the only way that this can happen is if this segment has been integrated into the chromosome of the target site. So in this case, you see that homologous recombination happens at much better frequencies and much more efficiency, because the cells are able to recognize under these circumstances linear DNA, and then the recombination reaction is mediated not by the Rec-A system that requires big segments of homologous DNA, but by small sequences that you have at the end of the PCR fragment. So if you want to have a deletion, then the DNA fragment has to be engineered in such a way that the antibiotic resistance can be later be eliminated through another round of recombination that eliminates the intervening sequence between the two sites recognized by the recombination system. Okay, so it's is a so-called site-specific recombination between this sequence here and this sequence here, and then you have this kind of semi-homologous recombination mediated by a special recombinase that at the end enables the possibility that if you want to delete this B gene here between A and C, then you just generate this fragment that is flanked by these short homologous sequences, and then you have this integration, and then later you can eliminate what you have in between through another round of recombination. Well, this may look, again, complicated, but believe me that this simplifies things a lot. So the proteins that make the trick are proteins encoded by the bacteriophage lambda, and it involves 3 proteins that I mean the details of how they work at this point are not interesting, but what they do at the end is delivering a strong recombination activity with very, very short segments of DNA, and in that respect they do much better than other homologous or bases for recombination based on homology or extensive homology. Well, these are some details on how this red system works, and well, there are basically 3 enzymes. One of them degrades the double-stranded DNA in one of the streams, and then another protein called beta then enables these streams to be protruding and therefore being able to attach to the corresponding DNA in the target molecule, and at the end the system works well. Well, there are details that perhaps are not so interesting. I'm having problems with I mean, you know, the text. What happens with the text here? So maybe in the break we can fix this a little bit. Okay, so for this type of recombinering you need various conditions, but in any case, I mean, this paper where the technology for this recombinering with double-stranded DNA worked had many say citations, and it works quite well. However, if you remember what I mentioned before, still this system enables you to use, I mean, to enter mutations one at a time. So despite the fact that every mutation takes shorter than the previous method, the reality is that you cannot multiplex it, okay? So what to do next? Well, the other, the possibility came to make oligo-mediated allelic replacements through recombinering, but instead of using double-stranded DNA, to use single-stranded DNA. So recombinering in E. coli works with oligos and one of the proteins for recombination, but you need to have single-stranded DNA. So in this case, what you do is the following. So when bacteria replicate, they form what they are called replication forks, like here and here, and then transiently the 2 strands of DNA separate, and then in some cases you can bring in an oligo from the outside that will invade the replication fork, and will replace what are called the Okazaki fragments, namely the intermediates that are there for DNA replication. Okay, so the DNA replicates, the 2 strands open, but then if you add a lot of single-stranded DNA that has homology to some of the regions that are being replicated, the single-stranded DNA will invade that replication origin and that replication fork, and therefore you will inherit the sequence that is in the oligo that you add to the system. And this process is facilitated by a recombinase, by a protein that is called beta, because it stabilizes single-stranded DNA that you add from the outside, and at the same time it will facilitate the entrance of this complex in the replication fork. So that's something that has been known for some time. So this is exactly what happens. So this is the replication fork. So if you want to mutate one particular target here, what you do is that you treat the cells with this mutagenic oligo, where you have a mutation there, together with this recombinase that can be expressed in vivo. And then this oligo will pair with the target in the genome, obviously will not be a perfect pairing because it will have a mismatch or more mismatches, but at the end it will be able to be inherited because this oligo will be recognized as an Okazaki fragment, one of these replication intermediates, and will be inherited, okay? So conceptually, I think that this is much easier to understand, that if you want to enter a specific mutation in a place of the genome, then you prepare an oligo that carries that mutation, and then at some point, while cells are growing, you invade the cells with the oligo, and the oligo will find its way to its target, and then the mutation will be inherited. Okay, so this is what is called oligo-mediated allelic replacement, and is very easy to do from a technical point of view, and the frequencies are very, very high. And they can go between 10 to the minus 2 to 10 to the minus 5. 10 to the minus 2 means that even without any external selection, 1 out of 100 colonies will carry the mutation. And this is something that is very useful because 100 colonies is something that you can handle yourself with no problem, okay? And you can say, well, this is good enough, but still we may deal only with one single target. What if you want to, say, implement many different mutations at the same time? Okay, the solution is this wonderful method that I'm going to explain you in some detail that is called MAGE, okay? Do you know what MAGE means? Have you ever heard the term MAGE before? Okay. Okay, obviously, MAGE in this case is not Make America Great Again, is multiple automated genome engineering, okay? So in this case, the name indicates, is a technology that enables to enter simultaneously many different mutations in a given genome, okay? And that's something that is really, really cool. Okay. So this method was published in the I think it was just before the 2000s, and this is a paper that is a landmark paper in the history of synthetic biology, because as I just mentioned, it enables to enter many different mutations at the same time in the same genome, okay? And therefore it enables a large-scale programming, and in some cases evolution of cells, because it targets many locations in the chromosome of a single cell and produces, if necessary, a good combinatorial genetic diversity. Because imagine that you want to enter 200 mutations at the same time, but you are not sure which of the ones that you have there are going to be better for your purposes. Okay, well, I hope that in 1 hour our technician can help. Well, anyway, so in this case, what we have here is that the population of cells, instead of being transformed with just one oligo, is transformed simultaneously with many mutagenic oligos, okay? Like here. So you have the cells, you are in point 1, then you induce expression of the recombinase, then you chill the cells, you wash away, you add the oligos, you deliver the oligos through electroporation, you recover the cells in media, selective media, and then you grow them again, and then you take a look to the phenotype that you're interested. And if the phenotype has not developed completely, then go over and over and over and over and over. And as it happens that in every selection in every selection, say, cycle, about 1% of the cells incorporate the mutation, then if you have sufficient selection and incorporation cycles, at the end you have a large population of bacteria that will have incorporated all the mutations of interest. So in fact, this can be, or theoretically, it can be automated, and in this original paper, the authors claim that you can have a machine that will run the cycles simultaneously without having to intervene from the outset. So the tricks that are behind this possibility of making many mutations at the same time is that it is possible to make conditionally express protein B, that is the recombinase that recognizes, that enables the incorporation of oligos into the replication fork, because it protects the 3' ends from the degradation of exonucleases. So you take bacterial cells and you transform just PCR, not just PCR, but single-stranded DNA, then that DNA will likely be destroyed because of the nucleases, okay? So by expressing this protein called the beta protein, then the single-stranded DNA is protected from the action of exonucleases. And also, in some cases, there's a chemical trick to further extend the life of these single-stranded DNA oligos, because the ends of the oligos are modified in such a way that they also become resistant to the action of the nucleases. So what is the advantage of MAGE versus other previous approaches? Well, the best of all is that it's highly scalable, it can be multiplex, and it operates continuously in vivo. Okay, so these are advantages in respect to homologous recombination that I mentioned at the beginning, and also this recombination with PCR fragments that I also told you. Okay. Well, and this is one of the pictures that the authors of the paper show about the robot. I really don't know how well this works, but you know, the idea is that you have a cyclic operation for making this type of mutations. Instead of having you doing the trick, then everything can be done, say, by a machine. Okay, so in this paper that as I said, is one of the a big landmark in the history of tricks and methods for genomic engineering, then it represents the number of MAGE cycles and the number of times that you can enter different mutations in the genome. Okay, so you know that you see that in 0, well, you have no incorporation. With 2 cycles, you start entering incorporations. By 5, much of the population starts getting incorporations, and by cycle 15, basically all the population has incorporated mutations. Okay, so that's something that is quite nice, and if you have a phenotype that you can follow, then things are easy to say visualize. So to make this story like more visual, so what developers of this technology propose was to convert E. coli in a bacterium able to produce lycopene. So lycopene is the product, the red color that, the product that endows red color to the tomatoes, and obviously E. coli is a bacterium that does not make lycopene. However, this molecule here, in principle, could be produced by E. coli if you enter in the genome of E. coli a very large number of mutations. Okay, so this is E. coli, and metabolic engineering, and we'll have a class on metabolic engineering one of the last days, will tell you that to convert E. coli into something that produces lycopene, you need to enter 24 different mutations, including deletions, including allelic replacements, including insertions, including different types of things. So how do you enter 24 different types of mutations? In some cases, the mutations are just about modifying a little bit the expression of some genes that are involved in formation of lycopene. In other cases, you have to knock out some genes, okay? So, and you have to do that at the same time, and also in some cases, you know exactly how the different combinations of mutations will influence or not the outcome of the phenotype that you are after. Okay, so let me go before. Okay, so what these people did was to say enter all the mutagenic oligos that results in different mutations, and then just wait, and you know that after time, after different periods of cycling, of exposure to mutagenic oligos, then the E. coli starts to accumulate and accumulate and accumulate lycopene until it reaches a plateau that indicates that probably you have generated an optimal situation in terms of the mutations that are necessary for the formation of the product. Okay, so this is one of the many applications of this system, and maybe I will return to that subsequent thing. But other thing that you can exploit this technology for is, for instance, for entering many different mutations in the genome. For instance, for making a strain where you have edited a number of codons, you know? And well, this is another publication a few years later in which this match was combined with conjugation in such a way that one of the triplets could be changed in the whole genome of E. coli. Okay? So in this case, the mutation is not a mutation that will give you a color or will give you a clear phenotype. The mutations are about eliminating one of the codons that you have in the genome of E. coli, right? So as this system will have a very high efficacy for production of substitutions with the mutagenic oligos, and you can cycle the system as much as you want, at the end you can really get mutations all over the place. Okay, so this is what I told you, and you know, for instance, in this case, the idea was to replace all the stop codons TAG by the synonymous codons TAA, with the idea of liberating, of emancipating these codons here, or these stop codons, from being a stop codon into being a real, say, interesting stop codon, but different, and we can use that codon for other purposes. This is a term that is called emancipation. Okay, so emancipation means to take a codon and stop the meaning of that codon under its natural circumstances, and then reassign it for other uses, for instance, for encoding alternative non-natural amino acids. So that was what was made some time ago, and then this is a map with all the positions where every codon was replaced by using this technology. All right. So at the end, you can take all the codons of the genome, at least one of the codons of the genome, and by massively making these cycles of mutations that eliminate these codons, at the end you have a genome that basically is a erase of every single codon that you want to get rid of. Okay, so if you cannot do that at the same time, then you can go stepwise, divide the chromosome in various segments, and then apply this technology to each of the segments, and then at the end to recombine the different segments in a single bacterium. And this is what they did, and at the end they produced a bacterium that indeed happens without having the codon that we wanted to erase. Okay, well, this is very nice. But in some cases, what happens is that the frequency may not be sufficient, and when you have a situation where the phenotype is not clear, then the selection can be a problem, because if you want a variant of a protein, or you want to make a change in a codon, but you don't have a phenotype that you can select, that you can screen, then you rely on the efficacy of the mutagenesis. And even the best mutagenesis with single-stranded DNA may not be very high. Okay? So ideally, it would be great to have a counter-selection method that will be able to distinguish a wild-type sequence from a mutated sequence without having to rely on a phenotype. Okay? So how can you do that? Okay, then we open this big box of genomic engineering tools that are based on CRISPR-Cas systems. So CRISPR-Cas systems were, well, it has a long history behind. So first it was discovered that some bacteria have repeats in the chromosome. Then it was discovered that there was some proteins that could recognize these sequences and being able to cleave them. I mean, there's a long history behind that. I will not get into the details, but probably you will get the story in other subjects in your CV. But the idea and the basic facts that we want to exploit for the sake of this course is that CRISPR-Cas systems enable some, or provide some tools, some activities for RNA-guided editing of bacterial genomes. Okay. So, well, the basic facts about CRISPR systems is that they are, the name you remember, cluster regularly interspace short palindromic repeats, and they are the CRISPR arrays or CRISPR systems contain short DNA repetition. So you have 1, 2, 3, and each repetition is followed by short segments of spacer DNA that matches the sequences of viruses that theoretically the bacterium had been exposed before at some given time. So these CRISPRs are found in many bacterial genomes and are often associated to Cas genes that code for proteins related to CRISPRs. So under natural condition, CRISPR-Cas systems have to do with resistance to bacteriophage attack. Okay, so the idea in this case, and I'm just repeating things that perhaps you have learned in other contexts, is that when a phage invades a bacterium, then chances are that it will produce a specific a DNA. But if the bacterium has a memory of that segment of DNA as if it had been invaded before, then that was recorded before in the genome, and then the genome produces a RNA, and the RNA will bind to Cas9 and will create a nuclease that will attack the phage of the DNA of the virus. Okay, so this is a mechanism that is quite smart. Let's put it like that. But basically, for the sake of from a mechanistic point of view, the important part here is that you can have a nuclease that will specifically target one segment of the DNA of the virus, and that will be recognized on the basis that that specific sequence has been recorded in the genome of the affected bacterium before. Okay, so just telling the story very, very quickly. So, so this is how CRISPR works. So the DNA of the virus gets into the cells, and then there's a pre-existing RNA that matches bits and parts of the virus here, and then this RNA associates with proteins, with Cas proteins, and they form a nuclease that will kill the DNA of the virus there. Okay, well, some details here that are not so important. But in any case, so the important parts of the CRISPR system is that you have this array, then you have these repeats, you have spacers, you have Cas9, that is the nuclease that gets activated as a nuclease when it binds mRNA. And then for that to happen, you need to have a special sequence that is called the PAM sequence, that is the one that guides the whole complex to the DNA that you want to clip later. So it's a kind of blow-up of the essential component that we will be tackling today. So you have this Cas9 protein. So the Cas9 associates with a RNA that matches some specific sequences in the DNA, and the complex between this Cas9 and this specific sequence of RNA is what clips the DNA in a specific location. Or it's like you have a restriction enzyme that goes a la carte in a very dedicated way to a predetermined segment of DNA. Okay, so you have basically a nuclease that you can put anywhere in the genome. So this is what happens. This is the recognition, and then 1 second later, then the complex will cleave the DNA at that point, and the DNA will be destroyed. Okay, so that's something that is the basis. Okay, so this mechanism that under natural circumstances is good only for, or is good for just the defense of bacteria against phages, has been recruited by synthetic biologists and by other smart people to be converted into a phenomenal tool for gene editing. Because then by inserting a plasmid that contains Cas genes and specifically designed CRISPRs, an organic genome can be cut at any desired location. Okay, so that's a part. But then, on the basis of that, CRISPR-Cas systems have been reshaped as a tool for gene editing in various ways. Okay, so I'll tell you some of them. Okay, just for the record, there are different types of CRISPR systems. The ones that are more used might act as a system with 3 components. The 3 components are the Cas9 protein, then the so-called cRDNA, that is the DNA that guides the the protein, the enzyme, towards the target DNA, and then you have this tracing DNA that has to be incorporated into the system that enables the docking of this short DNA in the whole complex. But it is also possible to simplify things and then to generate what is called a guide protein, a guide RNA, in which we fuse these 2 components, these two RNA components, and then you produce a single RNA that is enough for doing the same job. So you don't have to have a separate tracing DNA, it can go something like that. It's a little detail. Okay, so this could be like the situation that we are after for cleavage. So you have this DNA that you want to cleave at one particular place, and then you generate artificially this guide RNA, and then the complex between the guide RNA and the rest of the protein will become a directed nuclease against something that you want to target there. So Cas9 can be produced as a protein, and then the guided RNA can be produced in various forms from a different promoter. It can be T7, it can be other types of promoters, and even in some cases, it can be produced from a PCR fragment with a T7 promoter, where you encode in the PCR fragment this guide RNA that eventually will convert Cas9 into a nuclease. Okay, so in this scenario, there are many things that you can do. In many cases, what you do is that if you want to enter a given mutation in a genomic location, and you know that the Cas9 complex will cleave the chromosome at a given place, then the only way to rescue the life or the viability of the bacterium or the biological system, because this is applicable to any biological system, bacteria, all the way to mammals, to mammalian cells, is to add what is called a donor repair DNA. So because of the recombination, then the area or the part of the DNA that is in between the two parts where you have homology will be incorporated into the genome here. Okay, so that's something that has been exploited in many, many different ways to repair specific sequences, to delete, to enter different types of mutation, because this is a kind of recombination. If you remember, in one of the previous tools, what we had was a cleavage by a restriction enzyme that we had incorporated a site for it in the place that we wanted to edit. In this case, we do the same, but the cleavage site is not fixed. We can fix it at our will because we have the possibility to cleave the DNA at a specific site there. Okay, so repair of this cleavage is what at the end brings about the changes in the target. Okay, so that's something that it was one of the very, very early possibilities to use this thing. Okay, so since then, CRISPR-Cas technology has exploded in many different directions, and in the IM competition, even from the very beginning, then people started to play with different projects in which you use the CRISPR system for making different possibilities, and maybe some of the projects that we will hear the last day will have to do with that. Well, you have different kind of funny stories about this. Okay, we're going to stop for 5 minutes, and then we'll continue with this because it's kind of fun to see. Okay, obviously we are getting deep into these technologies. I understand that they are following them are not the most fun thing on earth, but it's important that you kind of grasp, if not all the details, at least the idea that you have an entire palette of technologies to bring about specific changes in the genome of your favorite biological system. Okay.